Welcome back, everyone. Today we're going to be talking about experimental design. If you recall, the key difference between an observational study and an experimental study is that in an experiment, the researcher manipulates the conditions that some of the subjects are exposed to and leaves the other subjects unmanipulated as controls. Now, we perform experiments because observational associations can always be confounded by the influence of some other lurking variable. So, for example, we might think that some variable x has some causal impact on some other variable y, and those two might be associated with one another in an observational study. But it's entirely possible that some third variable called z here, which we'll call the lurking variable, is what is actually causing the association by having an influence on both x and y at the same time. And as a result of this potential for lurking variables to lead to associations, in an observational study, although we can establish an association between two variables, we can't determine whether or not the two variables are causally linked. So for example, we can find all sorts of what are called spurious or false associations between two variables. And so here we have a very tight correlation between the divorce rate in Maine and the per capita consumption of margarine. Or perhaps the number of people who drowned by falling into a pool and the number of films that Nicolas Cage appeared in. Now, obviously, these are silly examples of associations, but when we see other associations in biology that seem to make sense to us, it's all too appealing to imply that these associations are somehow causally linked, when in reality, the only way to tell the difference between these associations and spurious relationships is through the experimental manipulation of one of the variables involved. Experiments get around this problem of a lurking variable because the researcher manipulates the conditions that some of the subjects are exposed to, and as a result breaks apart this effect of the lurking variable and allows us to determine cause and effect. But if we want to use experiments to be able to tell the difference between correlation and causation, we need to design our experiments carefully. When designing experiments, there are two important things that we need to pay attention to. First of all, we need to pay attention to how we assign subjects to our experimental groups. And second, we need to pay attention to the methods that we use for applying our treatments. In an experiment, we need to use the same types of approaches that we used for determining which subjects were going to be included in our statistical sample. But in this case, we need the same approaches to determine which individuals will be part of which experimental treatment group. The important point being that each subject in our experiment needs to have the same chance of being included in each experimental group. Now, the easiest way to do this is through the random allocation of subjects to treatment groups. So when we randomly assign subjects to treatment groups, we break apart any associations between any lurking variables and the conditions that are being manipulated in our experiment. So let's say we had this hypothesized causal effect of variable x on our response variable y, but we were worried that some other variable z might be acting as a lurking variable and actually causing this association, rather than x having a causal effect on y. Well, to break apart this effect of z, we could experimentally manipulate the level of x rather than relying on observational variation in x and its association with y. Now, by experimentally manipulating x, we don't eliminate the effect of z on y, but we break apart this association between z and x. And this ensures then that variation from lurking variables is similar in our two experimental levels in our manipulation of this variable x. So let's walk through a little bit more of a tangible example of what I'm talking about by a lurking variable. It's a common finding in ornithological research that birds that lay their eggs earlier in the season have higher fitness. But is this association between lay date and fitness actually causal, or is it influenced by some other lurking variable like body condition? Birds that have access to more food or are in better body condition lay their eggs earlier and have higher fitness. And so it's entirely possible that this association between when a bird lays its eggs in the spring and what its fitness is entirely determined by this lurking variable, such as body condition. So the way to determine then whether this association between lay date and fitness is actually causally related could be determined by experimentally manipulating the date on which females lay their eggs. So if we experimentally prevented females from laying eggs for, say, a 2-week period and compared the fitness of those females that laid their eggs 2 weeks later to those control birds, we could determine whether this relationship between lay date and fitness was causal. And the reason we can do that is because that experimental manipulation of lay date breaks apart this association between body condition and lay date through our experimental manipulation of lay date. Now, body condition is still going to influence fitness, but it's no longer going to confound the relationship between lay date and fitness because we've broken apart this association between body condition and lay date by experimentally manipulating lay date rather than relying on natural observational variation in lay date, which might be influenced by something like body condition. So in addition to paying attention to how we allocate subjects to treatment groups, we also need to pay careful attention in experiments to what it is that we actually manipulate in our experiment. As a general rule of thumb, it's really important to pay attention to what the entity is that you want to draw your conclusions about, and to perform your manipulations and apply your treatments at the level of biological organization that corresponds with the thing you want to draw your conclusions about. So if, for example, you want to draw your conclusions about individuals, then you should randomly assign individuals to treatment groups and apply those treatments to individuals. If you want to draw your conclusions about ecosystems, you should randomly assign ecosystems to treatment groups and apply your manipulation at the level of the ecosystem. Now, this isn't always possible, but it's important to keep this rule of thumb in mind if we want to try and have the best experiments possible. So when you're performing an experiment then, decide what it is you want to draw your conclusions about and replicate and manipulate that thing. If you want to draw your conclusions about populations, then replicate and manipulate populations. If you want to draw your conclusions about individuals, then replicate and manipulate individuals rather than manipulating groups of individuals or populations. So for example, I was interested in testing the effects of food abundance on population density in red squirrels. So as a result of my interest in populations, I replicated and manipulated entire populations of squirrels. So in this experiment that we ran across 14 years, we had 3 control populations and 3 experimental populations where we provided every individual in our experimental populations with supplemental food throughout their entire lives. In addition to paying attention to what we should manipulate, we also need to pay careful attention to how we perform our manipulation. Let's say we were interested in testing the effects of exercise on stress levels. So imagine then that we randomly assigned executives into 2 treatment groups. In one treatment group, executives were told to exercise for 30 minutes per week. And in the second treatment group, the executives were told to exercise for 30 minutes each day. So as a result, then, we have manipulated the amount of exercise that those executives are performing. And this is better than an observational study, where an observational association between exercise and stress might be confounded by some other lurking variable. But in this experiment, our executives were aware of whether they were in the low or the high exercise group. And as a result, this knowledge of the treatment that they were experiencing could influence their response to the psychological tests designed to measure stress. The solution to this problem, then, is to apply our experimental treatments in a way that the subjects are unaware of the treatment group in which they are involved. And this is referred to as blinding. And a good example of this is the use of placebos in drug trials. So let's say we were interested in testing a research hypothesis that a vaccine will make subjects less susceptible to the common cold. In addition to randomly assigning some subjects to receive the vaccine capsules, we would then also need to have our subjects that were assigned to our control group to receive a placebo that otherwise looked exactly the same, but didn't have the active vaccine within it, in order to make sure that our subjects were unaware of which treatment they were being exposed to. And this is really important because placebos have been shown to have a very positive effect on many aspects of psychological and physical health, where we might expect subjects exposed to the placebo to report lower rates of experiencing colds, even without any active ingredient in that placebo treatment. Even in cases where our subjects are unaware of the treatments that they're receiving, we still need to make sure that our experimental manipulation doesn't have any inadvertent effects on our response variable. So, for example, in this study of long-tailed willowbirds, the researchers were interested in testing the effects of the length of a male's tail on his mating success. And instead of relying on observational associations between the size of a male's tail and his mating success, the experimenters experimentally manipulated the length of a male's tail by cutting their tail feathers back to a length of 14 centimeters. Now, this changed the length of their tail, but it also included a physical manipulation of the birds themselves. And as a result, the experimenters were careful to include in their control group birds that had their tail feathers cut, but then glued back on to control for any influence that the cutting or handling might have had on the birds. As a result, the only difference then between the experimental birds and the control birds was that the experimental birds had shorter tail feathers than the controls. And this manipulation of our control group in a way that is methodologically identical, but is missing the key ingredient or the key hypothesized agent, is what's referred to as a sham manipulation. And we'll talk about sham manipulations a little bit more in another video on experimental pitfalls. Now, as an example of both some good and some bad practices in experimental design, I'll briefly explain the research that I performed as a master's student, where I was interested in testing the hypothesis that breeding by young of the year female deer mice is limited by dietary protein. So in this particular hypothesis, then, I'm interested in individual mice. So that is the biological level that I want to draw my conclusions about. So proper experimental design then would involve replicating and manipulating individual mice. Now, there were two ways in which this experimental design was not ideal, and one way in which I think I did a pretty good job of designing this experiment. First of all, in this experiment, my treatments were not assigned randomly. I did this experiment in the context of an ongoing long-term study of mice in Kananaskis, Alberta, and as a result, the long-term control populations could not be manipulated. And so I wasn't able to randomly assign treatments to populations because I couldn't manipulate these long-term control populations. Okay, so this experimental design was not ideal because I didn't assign my treatments randomly. The second way in which this experimental design was not ideal is because my question was at the level of individual mice, but I wasn't able to manipulate the availability of food to individual mice because I couldn't feed individuals without having the food be available to other individuals in the population. And as a result, even though my question was about individual mice, I manipulated the availability of food accessed by entire populations. So some populations were control populations, some populations received a supplement of high protein food, and other populations received a supplement of high energy but low protein food. Now, the fact that I had these two experimental treatments and one control was the aspect of the experiment that I think I did pretty well. My hypothesis was about the effects of dietary protein, but you can't supplement an individual with dietary protein without also supplementing them with calories. And so, as a control for this caloric supplementation, I also provided mice with access to high energy but low protein food. And the results of this experiment are shown in this figure on the bottom, where it did appear that breeding by young of the year female deer mice was limited by dietary protein, because when I supplemented them with high protein food, a much higher proportion of those females were able to breed as young of the year than females in control populations or the high energy but low protein supplementation. And so this experimental manipulation of high energy but low protein food told me that the protein supplementation had its effect not because of the energy that was provided, but perhaps because of the protein itself, which was the main difference between these two food types that were supplemented. So in scientific experiments, then, the researcher manipulates the conditions that some subjects experience while other subjects remain as controls. And this type of experimental design, then, is really useful for being able to determine causation in an observed relationship rather than just an association. In an experiment like this, it's important that we assign our treatments randomly so that each subject has the same probability of appearing in each of our different treatment groups. And if we do this, we can break apart any associations between our hypothesized explanatory variable and our response variable that might be caused by these unmeasured lurking variables. But in performing these experiments, it's important to think carefully about what it is that we want to draw our conclusions about and to manipulate and replicate those things.